Hello everyone, in this video I'm going to talk about gene editing technologies. How does it work after so long time of here? At least almost about 3 months, 3 days I'm recording this video of technologies. Otherwise I'm recording random videos at all. I'm recording only random videos. But now I am back. I am back. You feel sometime low in your journey. When you feel low in your journey, it doesn't mean that you have to keep quit everything. Then also you have to keep moving in your journey. Then only you can achieve your goal. Otherwise you will got stuck out to be there. You have to make a plan out in order to bounce back again. Are you getting what I am saying? So in order to not get your work down, you just have to keep moving. Either it could be a slowly, or it can be other, like do some kind of the do some work related to that stuff you are doing. Either it can be of itusa either it can be in a little bit. That will be keep keep what keep what keep what it will be keep rearrange the change that we have made from previous stuff. It will not able to break down that stuff, that chain that you have processing or building from quite long period of time. That what I am saying. Are you getting what I am saying? Are you getting what even I am saying? Are you getting? 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 So what I am going to do? What I am going to do? What I am going to do? Today we are going to talk about very exciting technology. That is gene editing technologies. World Cup is also coming. We have to cover all that stuff also for almost 2 months. That's going to be a quite exciting time, bro. That's going to be a quite excited time. How I am going to do it, I don't know, but I'm quite excited about it. I'm going to do commentary out World Cup for almost 2 months. 48 match out to be there, 48 match out to be there, one match at a day. We have to cover all that stuff. I think it will start from 5 of October and will be gone till when? Will be gone till, I think, 18 November. Yeah, I think so. Yeah, we have to cover all that stuff, bro. We have to cover all that stuff. I'm quite excited about that. I'm going to. I'm quite excited about that, bro. Let's see how this goes. If we able to do or not. I was thinking to do it from quite bit of time. Like I have decided to cover IPL, upcoming IPL match. What, what? What due to many conditions or minor stuff I able to not do due to my what? What can we say that? Due to my inferiority or self-doubt, I have not able to do that. But this time I'm quite excited. Let's see how it goes. Let's see how it goes. Let's see how it goes. So we have to cover today what? We are going to cover what? I'm going to cover what? I'm going to talk about talk about talk about I'm going to talk about what I'm going to talk about, bro. Gene editing technology. We are to talk about gene editing technology. We are going to go how does that gene editing technology to work? All that stuff we are going to cover in this video. Let me done it. Let wait a second. I'm going to start in a minute. Just wait a second. It's look good. It's look good, bro. Not that bad. Even more videos or more stuff we're going to cover. Not that only. So gene editing technology refers to the group of advanced biotechnology. One, reading that stuff or word itself, it seemed too scary. You can edit your genetic material also. That sound very scary. Gene editing technology refer to the group of advanced biotechnological method that allow scientists to modify the DNA of organism include humans. Yeah, this technology enable precise and targeted changes to the genetic code, opening up possibilities for treating genetic disease, understanding fundamental biological process, and even enhancing traits in plants and animals. So this is stuff we can do with gene editing technologies. What can we do? We can modify DNA of organism are including every kind of species out there, which include humans also. The technology enable precise and targeted change in genetic code. Even that the specified goal we have assigned in order to do in a genetic code, we can do also that. Okay, opening up possibilities for treating genetic disease, understanding fundamental biological process, and even enhancing traits in plant and animal. We can saw different type of plants out to be there, like in modified version of different kind of like experiment has done in order to do so. Maize is kind of like that. Maize, Makkah, if you know that about that, that has been a modified plant that we have possibility that have possible due to the genetic enhancing or genetic process. I think so. I have read about that. Like that's why I'm quite sure sure about it. I've read about it somewhere. We have covered, I think, CRISPR Cas9 already. What we have done? What is it? What is it? Now we are going to cover about when it's point about gene editing technologies like CRISPR-Cas9, other gene editing techniques, application in medicine, genetic engineering and agriculture, ethical and regulatory concerns, off target and way future possibilities. All this stuff we are going to cover in this. Okay, okay. We're going to cover various points around the gene editing technologies, how that is work, like CRISPR-Cas9 and other gene technologies. Then in the last, we're going to cover around 8 questions related to gene editing technologies. Are you ready for that? 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 Yeah, CRISPR-Cas9. What is CRISPR-Cas9? How does it work? Gene editing technology. CRISPR-Cas9 is a revolutionary gene editing system. Uses RNA molecules to guide Cas9 protein to specific DNA sequences. Cas9 that cuts the DNA at the targeted location, allowing for gene modification through repair mechanism. Is it possible to do so like that? Hmm. Second is other gene editing techniques. Gene editing techniques. What other technologies has been there in order to do so? Question apart from Cas9, what are the some other gene editing techniques such as zinc finger nucleus transcription activator like effector known nucleases? It's nucleases. Tell us how do you compare the CRISPR gene and are the old gene editing method that has also created target DNA breaks for modification CRISPR-Cas9 has been gained widespread popularity due to its simplicity, efficiency, and versatility. Application in medicine. How does the gene editing technology is being applied in the middle of medicine? What are the potential benefit and ethical consideration? Gene editing hold promises of gene. Why does it? They are like a question answer. We are not here to cover question answer. We have to cover out like different part of the genetic technique. I have not keep in mind while creating this slides. Sorry for that. From next time onward, I'm not going to do this kind of mistake. I'm here to talk about different part, not in form of question answer. Question answer we have to cover in other part of the slide, not genetic engineering. This also in a question answer format. How gene editing technologies, I'm considering that the question that we are discussing now not to be in coming slide that we have to cover in question answer format usually. If it is so, then it will be bad for our video. Then we are not covering some great amount of questions out there. Then we are repeating some stuff again and again. Okay. Okay. Are you ready for that? How gene editing technology is being used in agriculture, such as in the development of genetically modified crops, and what are the implications of food production and sustainability? Gene editing can create crops with desirable traits like drought resistant and improved nutritional content. It is potential to enhance and security and reduce the environmental impact of agriculture. Ethical and regulatory concerns. What are some of the ethical dilemmas, regulatory challenges associated with gene editing in humans and other organisms? Ethical concerns include the potential for designer babies and intended genetic consequences. Regulatory changes involve determining safety and oversight of gene editing experiment. Off target effect. How does a scientist address the issue of off target effect in gene editing, where unintended genetic change can occur? What are the methods used to improve the precision of gene editing techniques? Researchers work on the improving of specificity of gene editing to like reduce off target effect through various methods like protein in engineering and RNA optimization. Future possibilities. What are the some future possibilities of gene editing technologies such as gene therapies, synthetic biology, another potential form of editing trait beyond disease-related genes? Answer: gene editing could be revolutionized healthcare by providing treatment for currently incurable disease. It also opens the possibilities for creating synthetic organisms and enhancing traits in humans, which raises ethical and societal questions that we are going to cover various questions. What are the key differences between the gene therapy and gene editing? How does this approach impact the treatment of gene edited disease? Gene therapy involves adding, replacing, reappearing gene with patient cells, while gene editing directly modify the patient gene. Gene editing offers precise control over genetic changes and potential for permanent corrections, whereas gene therapy may involve inserting functional copies of genes. How do scientists tackle the challenges of delivery when gene editing technologies of therapeutic purposes, especially in context of vivo application? Delivery challenges involve ensuring that gene editing tools such as CRISPR-Cas9 reach the target cells and tissue in the body. Researchers are developing innovative delivery methods, including nanoparticles and viral vector, to enhance the efficiency and specificity in the vivo gene editing. In the recent year, base editing and prime editing have emerged as advancement in gene editing. Can you explain this technologies, these advantage and potential limitation? Base editing allow the precise conversion of one DNA base pair to another, while prime editing enable insertion, deletion, and substitution of genetic sequences. These technologies offer greater precision and fewer unintended change that traditional CRISPR-Cas9, but also still evolving, and their application may be context specific. What ethical consideration arise when apply gene editing to enhance traits in humans such as intelligence or physical abilities. How can society navigate these complex ethical dilemmas? How do gene editing contribute the field of synthetic biology, where researchers aim to create novel biological organs with specific function? That's one of the mirror. Beyond healthcare, what does it have for? Does it affect our video? No, it's not. I think 15 minutes has passed in this video. Yeah, we have got various type of questions in this slide itself. Like what? How do regulatory bodies or international agreement address the global governance gene editing technology, considering the potential for cross-border research and application? What recent breakthrough on emerging trade in gene editing technologies hold the most promise at recent breakthrough include advancement in base editing, prime editing, and epigo epigonomy editing, which offer greater precision and fewer side effects. These technologies hold promise for treating a wide range of genetic disease and conducting fundamental research in genetic and epigenetics. Okay, that's it for the video. We'll see you next time. Till then, keep making videos. Okay.